I'm Brian Cottle. I'm a machine learning and biomedical engineering postdoc working with SandboxAQ on the AQMed team, which is building a magnetic cardiography device. We're going to talk a little bit about some of my research that I did in academia, which was really relevant for me as I pursued my career and ended up at SandboxAQ, because my research was focused on applying machine learning, more like custom machine learning algorithms on custom data sets, proprietary data sets, to cardiovascular engineering. This particular research is on building 3D models of the cardiac conduction system in healthy neonatal human hearts. We'll talk a lot about what that means, why this was important, the implications, how we did it, most importantly, and then what came of it. Getting into some of the background behind this research, if we start off with the cardiac anatomy, the anatomy of our human hearts. We all have these hearts. They all have generally a very consistent anatomy, which involves, at a high level, 2 atria and 2 ventricles. These are chambers in the heart that allow blood or pump blood from the body into the heart and then back out either to the lungs or to the rest of the body. So we've got atria at the top, which gather the blood, and then the ventricles, which pump the blood out away from the heart. Got a cute little diagram here showing us what that flow normally looks like. However, in some cases, infants can be born with congenital defects. These involve changes to the anatomy of the heart that affect the function of the heart. On the left, we've got our example of a normal heart. On the right is an example of something called tetralogy of Fallot, which just has a variety of different abnormalities in the heart. We've got smaller vessels, we've got different valves, we've got a hole between the 2 ventricles, which allows blood to mix, which shouldn't be mixing, some thickening of the walls. Anyway, there's a bunch of changes which affect the functioning of the heart and subsequently affect the flow and how efficiently you can get the blood that you need either to your lungs or to the rest of your body. Causes all sorts of problems. In addition to, we got our typical anatomy with the atria and the ventricles. Another important component is the cardiac conduction system. So these are specialized tissue that reside in the heart, in the muscle of the heart. Fancy names like sinus node, the AV node, bundle of His, left and right bundle branches. What's important about these is that they're inside the muscle. They're specialized muscle tissue that regulate the beating of the heart. This is, it helps the heart, beat with the right rhythm, with the right speed, in the right, sequence where the top beats before the bottom. And anyway, to a very, very precise degree, this cardiac conduction system controls the heart. When we have hearts with congenital defects, not only is the general anatomy and function of the heart affected, but this cardiac conduction system is also moved around inside the heart and affected. Particularly the AV node shifts in its location. Because we've got this example here where we've got a hole between the 2 ventricles. The AV node can move around as well as the left and right bundle branches. Now, corrective surgeries, when we want to help patch a hole or fix some of these congenital defects, oftentimes require lesions and sutures to be placed very, very near or in almost the exact location of the sinus node, and in very particular, the AV node, the bundle of His, and left and right bundle branches. What's difficult is that in these hearts, when surgeons are performing these surgeries, these corrective surgeries, the nodal tissues are hidden inside the muscle of the heart. They don't know exactly where they are. Not only that, but for comparison, think about the adult human heart is about the size of your fist. As you hold that up, that's how big the heart is as it sits inside your chest. Now, these infants' hearts are no bigger than a strawberry. So these tiny little hearts are being operated on in open heart surgery, and the surgeons are tasked with using their extraordinary knowledge and experience to correct these defects while also not harming the cardiac conduction system. Sometimes harm does come to the cardiac conduction system, which introduces things like conduction abnormalities, arrhythmias, inefficiencies in the heart, sometimes failure for the heart to beat properly entirely. This damage ends up requiring these patients to have pacemakers implanted so early in their life, within 1 or 2 months of being born. that they need to carry that with them, that burden with them throughout the rest of their life. So another difficult aspect of this is that it's really hard to know exactly where these tissues are, and they're really hard to study. From such a small population of congenital hearts from infants, it's hard to know the typical variability that we can expect in the locations for the sinus and the AV nodes. So to help with this, our research team developed a method for creating really high resolution 3D models of the cardiac conduction system tissues, the sinus node and the AV node, in order to help us understand what the baseline variability is for healthy individuals, for healthy infants. So we could know, well, we usually expect the AV node to be here or this deep or to span this length or width or thickness. And so to do that, we partnered with Boston Children's Hospital and a few other companies to gather donor tissue. So these are donor human hearts. From those, we would extract the sinus node and the AV node. So we've got our little sample with the AV node inside it. We would section these tissues at about 25 micrometers between sections. So we get these really, really thin, 4 micrometer thin sections of tissue, which we then use typical histological methods to stain them. So we get some colors, some bright colors that pop out and show us where the muscle is, where the connective tissue is, where the nodes are, where the vasculature is. And so with that, we ended up with a very massive data set, more than a terabyte of images. And then we used some custom machine learning to segment out these regions, the regions of interest, whether it's muscle, whether it's connective tissue, the vasculature, the neurons that innervate the sinus and the AV nodes, and the nodes themselves. And we've got an example here from this paper, one of the figures that shows the results of some of this segmentation. So in some of these figures, we've got the stained tissue on the left and then the segmented tissue on the right, where we've got the light blue is the node, the yellow is the different vasculature, and there's some other stuff in here, but essentially this gives us a really high resolution description of precisely where the muscle is, where the nodes are, where the vasculature sits, and how much vasculature is near the nodes, how much is further away. Because another issue is that when surgeons perform these surgeries, maybe they miss the node, did a good job at missing the node, but again, it's really hard to know the composition of these tissues during surgery in such tiny hearts. And maybe they accidentally nick or place a suture in some important vasculature that provides blood to the nodes that then ends up causing issues. So with this segmentation, we then were able to take all these hundreds of sections of tissue these hundreds of sections of the tissues per tissue sample, and stack them again and use a lot of other advanced computational methods to reconstruct them into 3D models like this one here. So this example here is of the sinus nodal region. We could go into detail about the very precise anatomy and the locations of this tissue, but some of the important things that we can see are we can see how much vasculature is providing blood to the sinus node, where that vasculature comes from. Additionally, where the neurons are in relation to the sinus node, and how deep the sinus node is from both the outside and the inside of the heart, how thick it is, how long it is, how deep it is. So we can precisely quantify these things down to about 5 to 10 micrometers throughout the entire piece of tissue here. I've got another example here that we created of the AV node. Again, we could go into more detail about the AV node, but this is about the anatomy. But shows us how precisely we can quantify where the AV node receives the majority of its blood, but the majority of its blood supply, how big the left and right bundle branches are. So these are little branches that come off of the AV node here on the side. How different the branches are. So on the left side, we've got something called the left bundle branch, but actually is less of a branch and more of a sheet, which is something that we were able to confirm in these neonatal hearts, as well as how much innervation there is provided to the AV node. From these, we can quantify pretty precisely. Again, like I mentioned, we've got the depth, we get the vasculature. Here we've got some examples showing the depth from the tissue surface in the AV node, and here again in the sinus node. And got a lot of really cool stuff from this. A lot of things that we could quantify and provide, and knowledge that we could provide to surgeons to help them with their surgeries. So this particular research leaned very heavily on custom computer vision models. And these are some things that very similar stuff we use at SandboxAQ and the AQMed team. We build very, some of these complex and custom computer vision models in order to help us analyze or extract data from very large data sets. And this particular example is one that I'm super excited about, that I really loved, because we were able to use multiple models in conjunction with, so we used a UNet in particular, we'll talk about that. We also used some tree-based segmentation to help us with really, really small and fine segmentation so that we could get large features extracted or segmented, like the vasculature, the nodes, but then we also were able to get down to the nuclei and segment individual nuclei from these tissues. So we have these massive models that have information that goes all the way down to the nuclei and all the way up to the whole, the scale of the entire model. Additionally, one of the things that I was really excited about, it was interesting to me, was that we had very high control over the entire pipeline. So we'll talk about this, how that affected this UNet, or a combination of a sparse segmentation practice, as well as ensuring extremely high quality labels and a very consistent imaging quality, gave us very performant models. We had control over exactly how the stains were applied, and we used machines to ensure that it was really consistent across every piece of tissue. We did our own imaging. We did our own registration of these images, so aligning them. We created our own custom interpolation methods between all of the slices to create these 3D models. In particular, if we talk about the UNet, this is a segmentation model which took care of the vasculature, the neurons, and a couple other important aspects of these descriptions of the composition of these models. That we created our own training set that described where the vasculature is, highlighted, segmented where the vasculature is in the heart. We did something called a sparse segmentation approach, which was where we took small sections of each image of the larger images, and in those small sections we made sure that we segmented, as we're annotating this data set, that we segmented the highest quality, most easy to describe or identify vasculature or neurons. And because we didn't do this exhaustively through the data set, but we picked and chose the best examples, and we made sure that our annotations were exquisite, very well done, if I do say so myself, because I was the one that did them, the model ended up performing better than our segmentations that we provided to it. It learned the representations of the vasculature of the neurons, so that it would. This is really funny. Is that eventually the performance, as we're watching the accuracy of our segmentations during the training process of this model, it got better, continued to get better, but then it got worse at the end as we trained. So there was a dip. And the reason that it got worse is because it was annotating the model was learning to annotate the vasculature that we hadn't even annotated ourselves. Like it had learned the representation so well that it was actually doing better than we had. So that was a really fun discovery, and something that we were able to put together for this project. So some of the takeaways here were that we are able, using this method, to quantify the nodal depth, the location, the size, a lot of variability very precisely. We were also able to identify, sort of discover, important variations in the location and the path of the AV node and its branches, where one of the right bundle branch would in some cases sit at the surface of the tissue, the inside of the heart, but on the endocardium. And sometimes it would dive into the ventricular myocardium more quickly. So we were able to identify these different variations. Again, the UNet that we put together surpassed our labeled dataset accuracy, using our sparse segmentation approach and very cleverly choosing what we provided to it. This enabled us to, to achieve very high quality segmentations on such a large data set with our, at the time, with our limited resources. And these methods now can be used and leveraged to study congenital hearts, or hearts with congenital defects, in addition to the healthy human hearts. So we have our baseline variability. We know the depth, we know the variability, we know the thickness that we could expect from a normal human heart. These methods can then be used to identify and quantify that variability in congenitally deformed hearts. And with that, I also want to make sure that we acknowledge the researchers and collaborators that helped us with this. Boston Children's Hospital, this was carried out at the University of Utah at the Cardiovascular Research and Training Institute. This research really got me on the path towards working at SandboxAQ, and in particular on the AQMed team, because there was so many different, this is even like a very small aspect of the research that we did. We also worked with and developed novel medical devices to help guide surgeons during surgery. So feel free to look up some of the other publications about that. This used light scattering spectroscopy, machine learning, and a variety of other methods. And so a combination of these things, this computer vision, working with massive data sets, creating custom quantitative models, these things really engendered in me like a love for working with very precise quantitative data sets. And that fits in perfectly with our work at SandboxAQ, where we're working not only with large quantitative models, machine learning, signal processing, developing novel medical devices, but also something that I really appreciate is that we are working on with the heart and cardiovascular engineering, cardiac electrophysiology. And this to me is extraordinary, and one of the reasons that I really enjoy working at SandboxAQ. And with that, I hope you enjoyed this tech talk, and make sure that you subscribe to the channel and check out some of our other videos.